Bismillah ar-Rahman ar-Rahim. This is the second practical immunology for first year, edited by Professor Dr. Batul Mudar Mehdi, MD-CHV, MSc, Fellow, Iraqi Commission Medical Specialization, Pathology, Consultant, Clinical Immunology. The third type of immune reaction is ligand assay, that we use substance binds with antibodies called conjugate, which is either fluorescent material, enzyme, radioactive material. The immunofluorescence test, we have two types, direct and indirect immunofluorescence test. The direct, we use the tissue section that contains the antigen and the antibody bind to it, and then we add the conjugate, which is the fluorescent labeled antibody, and this is used in the diagnosis dermatological disease like pemphigus vulgaris. And the indirect method, we use the tissue from the animal like hepatocyte or liver from the rat, and then we add patient serum that contain antibodies for the nucleus, which is for detection anti-nuclear antibody, for example, and then add the patient serum that contain antibody against the tissue, and then add the conjugate, which is the antibody labeled with fluorescent. And this examined under the fluorescent microscope, and we see the apple green color of the tissue, and for example autoantibody to thyroid gland, autoantibodies against the thyroglobulin or against the thyroid cell. The another type of the ligand assay using enzyme, which is called enzyme immunosorbent assay, ELISA, and this is used for detection antibody to HIV, antibody to hepatitis B surface antigen, HIV/AIDS, human immune deficiency virus, and here we use microtiter plate coated with the antigen, and then add the patient serum that contains the antibody, and we do wash to get rid from the excess antibody, and then we add the ligand and do wash to get rid from excess ligand, and then add the chromogene, and this leads to development of the color. And what happens? The principle is the antibody bind to horseradish peroxidase enzyme, and we add the substrate, which is H2O2, and this leads to horseradish peroxidase plus oxygen, and then this oxygen add chromogene in the reduced form, so this leads to for oxidization the chromogene and H2O only. This is wrong, no 2, only H2O, water, and this gives the color. And this is the reagent of the kit, and this is the microtiter plate, and give the yellowish color, which is indicate the presence of the antibodies. And this is the magnified microtiter plate. The another test is the radioimmunoassay using radioactive material instead of enzyme, and the same principle of ELISA and previously used for the hormonal assay, while nowadays used the ELISA test. The another test is Immunochromatography, which is a qualitative rapid test using a dipstick membrane coated with monoclonal antibodies against the antigen, while the conjugate is protein A colloidal gold conjugate used in many tests like detection of malarial antigen, pregnancy test, and other tests like the herpes simplex, like hepatitis, like toxo, like many tests used. This used as a screening test, and here used in the pregnancy if we have this is for the control and this is for the patient serum. If we have two tests, so this means that the patient is pregnant. The another test, which is complement fixation test, no more used today. It is used for detection antigen or antibody in the patient serum, and the oldest test in the immunity. And here we have two test tubes, and we have the antigen and add to it the patient serum. So this patient serum contain antibodies, lead to antigen antibody immune complex, and then add a rapid complement, and if this tube did not contain the antibody, so the antigen and the complement remain free, while if there is antigen antibody, so there is complement fixation, and then we add the indicator cell, which is the RBC coated with the antibody, and this leads to lysis and the release of hemoglobin and indicate negative and patient not infected, while here so there is intact AMBO receptor indicator cell, and mean that the patient is infected and had antibodies in its serum due to no cell lysis, while here there is lysis to indicator cell. Now we have the cellular immunoassay. We complete the humoral immunoassay. Here we have to separate the lymphocyte using a file called IPEC media, lymphocyte separation media with specific gravity of this media 1.078 used to separate the lymphocyte, and we get 4 layers: a plasma and platelets, mononuclear cells and lymphocyte separation media, RBCs and granulocytes. So we have 4 layers: diluted plasma, lymphocyte, ficle, red cells, granulocytes, and platelets. Platelets inside. Here, platelets in the lower layer with the granulocytes and the RBC. Here only the plasma platelets here with the granulocytes. Here the platelets in the bottom of the test tube, and use the lymphocyte for the enumeration of the T lymphocytes. Used for enumeration of the T lymphocytes. T lymphocyte had a receptor, CD2 receptor, and sheep RBC accumulate around it, and we enumerate how many rotating cells we get as a percentage of the number of the T lymphocytes. And also use, this is the rotating, this is the T lymphocyte and RBCs, sheep RBCs around it. Also enumeration of the B lymphocyte using the direct method at the conjugate, which is the fluorescent labeled antibody, and we get a bullet green of the cell wall of the lymphocyte, and this indicates B lymphocyte, while here the red one is negative. And also enumeration of T lymphocyte subset, T helper, T cytotoxic, using the indirect method, because this is the T cytotoxic, which is the characteristic of CD8, and add monoclonal antibody against the CD8, and then add a conjugate, and if it is positive, so give apple green color. And also we use it in the lymphocyte in the tissue typing, and we use the ficle and use the anti-sera, each tube against a specific allele, E37, CW2, 18, and we use a Terasaki plate, and then we use a Terasaki plate, and what has happened that this is the lymphocyte, and this is the HLA-B8, and add monoclonal antibody against the B8, and then add a rapid complement, and we add the trypanoside blue if there is if the patient had HLA-B8 on its surface, so there is rupture of the cell, and trypanoside blue enter the cell and give staining of the cell, and cell will be dark. And this indicates the patient had HLA-B8, and this is known as serological method, tissue typing by the serological method. And another method, the molecular method, using sequence-specific primer. Here we took the DNA from the patient, which is double-stranded DNA, and we use the PCR method, and the PCR increase the temperature into 95%. We do denaturation of a double strand into single-stranded DNA, and then we'll increase the temperature to 65, and then decrease the temperature to 65, and then we'll be annealing of the primer, and then we'll increase the temperature to 72 centigrade, and there is an extension of the DNA, and with multiple copies due to repeating of this process 30 times. And then we do a gel electrophoresis, and we get these bands which indicate and compare it with the table that contains the name of the alleles, and if it is migrating of the band, so there is name of the allele that patient gets it. And thank you for your attention.